Hello everyone. Welcome to the session of Anatomy of Flowering Plants NCERT Exemplar Part 2. Disclaimer. So learning objective, we will explore and understand the fundamental concept of Chapter 6 Anatomy of Flowering Plants by working through the NCERT Exemplar. So while solving the questions from the NCERT Exemplar, we will able to understand the fundamental concept from the chapter Anatomy of Flowering Plants. Index. So there will be some multiple choice questions, and that questions are from question number 10 to question number 18. Now in very short answer type question, we see there are question 6 to question 9, right? 4 questions are there, and short answer type question number 6 to 12. Long answer type questions, question 3. So total 20 questions are there. The first question is here. In which of the following pairs of parts of flowering plants is epidermis absent? This question is coming from the NCERT section 6.2. Let us see the options. A. Root tip and shoot tip. B. Shoot, bud and floral bud. C. Ovule and seed. And D. Petiole and pedicel. Let us see the answer of this question. So in which of the following pairs of parts of the flowering plant epidermis is absent? The correct answer is root tip and shoot tip. In the region of root and shoot tip, epidermis is absent. Root and shoot tip of an actively growing plant and has meristematic activity, right? That is the tip region where the meristematic cells are highly active. They are continuously in a division phase. So that shows the high meristematic activity at these points. The cells in this region are highly active and keep on dividing, right? So the cells do not get differentiated to the epidermal tissue because this is a type of tissue system, right? This is a type of tissue system, and here the cells do not get differentiated to form the epidermal tissue. So epidermis is absent in the root and shoot tip. Next question. How many shoot apical meristems are likely to be present in a twig of plant possessing 4 branches and 26 leaves? Let us see the option for this question. A. 26, B. 1, C. 5 and D. 30. And the correct answer is option C, 5. Let us see how it is. Apical meristem is present at growing apices. So growing apices are present where? At the apex of the plant. There are 2 apex, right? Shoot apex and root apex. So at these region, the apical meristems are highly active. They are present at that region. And here, according to the question, the plants are having 4 branches and 26 leaves. There is one main axis and the 4 branches. So every branch at the apex, they are having apical apices. Apical apices. The apex region that is continuously growing, that is continuously dividing. The 4 branch apex is having the cells that are continuously in a division phase. And you all know that apical meristems are present at the apices. So 5 growing apices are there, 4 at the apex of branches and 1 at the main axis. So total, the correct answer is here 5. So total 5 apices are there. Next question. A piece of wood having no vessels, trachea, must be belong to. Let us see the option. A. Teak, B. Mango, C. Pine and D. Palm. The correct answer is C, pine. And the pine, it is a gymnosperm. Pine is a gymnosperm plant. And the plant belong to the group that do not have vessel. Instead, they have tracheids. Okay. So this group is not having any type of vessels, but they do possesses the tracheids. That is one of the component of the xylem, right? So as we all know that the gymnosperm is not having any vessels. So here also, as a pine is asked in an example, so that is not having any vessels, but they do possesses the tracheids. Whereas the teak, mango and palm are angiospermic trees, right? Teak, mango and palm, these are some of the plants that are considered as an angiospermic. They are the members of angiospermic plant, and they do have vessels and tracheids. So angiospermic xylem contain vessels, tracheids, parenchyma and fibers, the 4 different components. Next question. A plant tissue when stained showed the presence of hemicellulose and pectin in the cell wall of its cells. The tissue represents, this question is coming from the NCERT section 6.1. And options are as follows: A, collenchyma, B, sclerenchyma, C, xylem, and D, meristem. Let us see the answer of this question. The correct answer is collenchyma. Explanation part. Collenchyma is a group of specialized cells that meant for the mechanical support. They are providing the mechanical support to the plant, and these are very, very special cells. So it is thickening at the corners of cells due to the deposition of pectin and hemicellulose. They are having the deposition of this pectin as well as the hemicellulose at their corners, right? Okay, next question is here. Fibers are likely to be absent in. So where the fibers are absent? This question is coming from the NCERT section 6.2. Let us see the option for this question. A, secondary phloem. B, secondary xylem. C, primary phloem. And option D says leaves. Let us see the correct answer. So option C is showing primary phloem. So fibers are likely to be absent in primary phloem. Let us have a thinking process for this particular question. Leaves have cells adapted for photosynthesis. Okay, so these cells are having photosynthesis, right? Leaves, they do possesses the mesophyll, and mesophyll cells are having a lot of chloroplast in them. So they are meant for the process of photosynthesis. Now, primary phloem forms in the apical meristem of the roots and shoot. Where this primary phloem is formed? It is formed in the apical meristem. It is formed in the apical meristem of root and shoot. Primary phloem has protophlloem and metaphlloem. These are the 2 components of the primary phloem, right? Protophlloem and metaphlloem. Protophlloem sieve tubes get destroyed as plant grows. As the plant is growing, right, the sieve tubes get destroyed. And other phloem cells that are converted to fibers, right? Now the next question is here. When we peel the skin of a potato tuber, we remove what we actually remove. NCERT section 6.2. This question is adapted from that section. And the options for these questions are A, periderm, B, epidermis, C, cuticle, and D, sapwood. Let us see the answer of this question. So A, periderm is the correct answer. Potato is an underground stem. Yes. This is the modification of the stem, right? Generally, we see the stem as a shoot part. But here in the case of potato, the stem is transformed into underground tuber, right? Underground tuber. And the outer epidermis layer of the stem is known as periderm. How can you say that the tuber is a stem? Because it is having nodes and internodes. You can see the eyes on that, notches from where the new buds comes out. So that shows that it is a stem. Now, the covering of this tuber of that you call as potato is periderm. That outer covering of the potato is periderm. So when we remove the skin of potato, what actually you are removing? That is the outer covering of the potato or a tuber, that is periderm. Next question. A vessel-less piece of a stem possessing prominent sieve tubes would belong to vessel-less piece of a stem that possesses prominent sieve tube. Two things are there, one regarding the vessels and another regarding the sieve tubes. Now, this question is coming from the NCERT section 6.2. Let us see the option for this question. A. Pinus, B. Eucalyptus, C. Grass, and D. Trocodendron. Let us see the answer of this question. So they see D. Trocodendron is the correct answer. Trocodendron, the species of these genus have a very unique feature of the angiosperm. Generally, the angiosperm is having vessel, but here you can see they lack vessel element. They are not having vessels in its wood, but has prominent sieve tube cells. They are having prominent sieve tube cells, but the vessels are absent in the trocodendron, and this is one of the exception in the case of angiospermic plant, right? Let us move to the next question. Which one of the following cell types always divide by anticlinal cell division? This question is coming from the NCERT section 6.2. Options are as follows. A. Fusiform initial cells, B. Root cap, C. Protoderm, and D. Phloem. Let us see the answer of this question. Okay, the correct answer is option C, protoderm. Anticlinal and periclinal division. Let us first understand this type of division. Then anticlinal division, the cells divide perpendicular to the surface of the plant body. So, if this is the surface, then like horizontal. Horizontal is the surface of the plant body and vertical is the division, right? The cells divide vertically. So this is showing the anticlinal division, right? Making a 90 degree at the surface. Periclinal cell division, the cells divide parallel to the surface of the body. So if you can see a surface of a plant body, then the section that divide the cells, it will also be the parallel to the surface. So that is showing the periclinal cell division. Now we will talk about the protoderm. Cells of the protoderm divide by anticlinal division. Here the cells of the protoderm that will divide by the anticlinal division. And the fusiform initial cells, root cap and phloem. These are the other options. Fusiform initial cells, cells of the root cap and phloem tissue divide by periclinal division. Periclinal division. But the anticlinal one we see in the case of protoderm. So the final answer is option C, protoderm. Next question. What is the fate of primary xylem in a dicot root showing the extensive secondary growth? This question is coming from the NCERT section 6.2. Options are as follows. A, it is retained in the center of the axis. B, it gets crushed. C, may or may not get crushed. And D, it gets surrounded by primary phloem. Let us see the answer of this question. The correct answer is option A. It is retained in the center of the axis. So primary xylem is present in the center of the root. Where it is present? It is present in the center of the root. And secondary growth occurs as secondary growth is occurring in the season, right? Then what happens? Growing season occur in the root of the primary phloem is pushed outside, whereas the primary xylem remains inside the root because it is toward the center. So once the cambium divide, it forms the secondary xylem toward the inner side and outer side secondary phloem. So phloem, primary phloem will be pushed as there are numbers of secondary phloem cells that push the primary phloem outside, whereas the primary phloem that will be pushed toward the center by the secondary xylem, and this is showing the secondary growth. Now we will have a very short answer type of questions. So the first question is here. What part of the plant would show the following? A, radial vascular bundle. B, polyarch xylem. And C, well developed pith. This question is coming from the NCERT section 6.2. Let us see the answer. Okay, let us see the answer. First, let's see the thinking process. Plant tissue show the distinct feature in the root, stem and leaves. Yes, we see the different types of cells and tissue arrangement in the case of root, stem and leaves. Because the thing that you see in the root will not be present in the stem, and the type of tissue that are present in the stem may not be present in the leaves. Phloem parenchyma is presently, but that is absent in the case of stem. Leaves are having the palisade parenchyma and spongy type of parenchyma that we do not see in the case of stem and root. So in that way, on the basis of the plant tissue, they are having the distinct features. Visual inspection reveals tissue origin, and when you see them, you can reveal what type of tissue and from where that is originated. Now, radial vascular bundle. Xylem and phloem exist on the separate root radii. When you see that xylem and phloem that are present on the different radii, that is showing the presence of radial vascular bundle, right? So this pattern is called radial vascular bundle. Next, polyarch xylem. Where you see this polyarch xylem? Multiple xylem strands from the polyarch condition in the monocot root. Yes, in the case of monocot root, you can see the xylem strands is more than 4. And 4 we see in the case of dicot, but in the case of monocot, it can be 6, it can be 8. So polyarch condition that we see in the case of monocot root. Well developed pith. Where we see the well developed pith? Parenchymatous cells form the pith in the dicot stem as well as in the case of monocot stem. The pith region is made by these parenchymatous cells. Next question. What are the cells that makes the leaves curl in the plant during water stress? This question is coming from the NCERT section 6.2. Bulliform cells. Bulliform cells are the bubble-shaped cells in the grasses. They are present on the upper surface of the epidermis, right? Loss of turgor pressure causes leaf to curl, reducing transpiration. So when there is a loss of turgor pressure, okay, this bulliform cells become flaccid. they release out the water from there, and this makes a pressure that makes the leaf to get curl, and in that way they reduce the transpiration rate. So here you can see the structure of the bulliform cells. These are the bulliform cells that are present at the epidermal surfaces, long elongated cells, right? So when they get filled, they become turgid. They become like a uh, the grasses. The grasses is having the very erect type of leaves. But once they are flaccid, they curl on to reduce the rate of transpiration. Next question. What constitute the cambial ring? This question is coming from the NCERT section 6.2. Let us see the answer. Cambium is the meristematic tissue. Yes, cambium is a type of meristematic tissue, and it is present where? It is present in the vascular bundle between xylem and phloem, right? So it is found in the dicot plant, and it is responsible for the secondary growth of stems and root. Yes, it is found in the dicot plant, and it is not present in the monocot. The cambium is not present in the monocot, and that is why their vascular bundles are called what? Their vascular bundles are called as closed type of vascular bundle. And just opposite to that, in the case of dicot, it is called open type of vascular bundle. So this cambium is responsible for the secondary growth of the stem and roots in the dicot plant. Interfascicular and intrafascicular cambia form the cambium ring. Now you see one thing, the cambium is just present in the between the vascular bundle, right? In the between the xylem and phloem. But what is that are present in between the vascular bundle? Two vascular bundle. They are having they are not having any cambium ring initially, but at the time of secondary growth. At the time of secondary growth, the cambium formation take place between the vascular bundle, and the continuous ring of the vascular bundle is formed, and that is called as cambium ring. They are formed by the meristematic activity of the cambium. So interfascicular cambium and intrafascicular cambium. Cambium between the xylem phloem, it is called as fascicular cambium. And between the vascular bundle, it is called interfascicular cambium. So at the time of secondary growth, the parenchymatous tissue that is present between the vascular bundle transformed into the cambium, and it makes a continuous ring of the cambium ring. So both combine to form a cambial ring, right? Next question. The cross section of a plant material showed the following features when viewed under the microscope. So when you cut a cross section of any plant material, whether it is a stem, root, leaves, whatever, the following features when viewed under the microscope. So let us see what are the following features. A. The vascular bundles were radially arranged. Where you see this thing. B. 4 xylem strand with exarch condition of the protoxylem. To which organ should it be assigned? Where you see these particular type of things. Okay, this question is coming from the NCERT section 6.1. Let us see the answer part of this question. So root is the organ which shows the feature given in the question. Yes. What type of root? Protoxylem. And metaxylem is there. Exarch condition they talked about. So this we see in the case of dicot root. Vascular bundles are present on the separate radii, and that is called radial arrangement. Means the xylem are present at the one radius, phloem will be present on the different radius. So alternate xylem and phloem bundles you see, and that type of arrangement that gives the radial arrangement in the vascular bundle. Protoxylem is toward the periphery of the root, thus making is an exarch condition. This is called endarch if the protoxylem is metaxylem is toward the periphery, then it will become the endarch condition, right? So this dicot root is the correct answer, because the features that belongs to the A feature and B feature which is mentioned in the question is belong to the dicot root. Okay, let's move to the next question. Short answer type of question. Here we are going to answer the short answer type of question. Question 7. Plant require water for their survival, but when watered excessively, plant die. Discuss on this. Very important question. This question is coming from the NCERT section 6.1. And this happened with sometime with us, like we water the plant with a compassion towards the plant, that we water more and more, 3 times 4 times in a day. Every day you are giving the water to your potted plant, and what happened? Ultimately the plant die. Then you say that I have taken so much care of this plant, but it die. Yes. So why it happened? This was our answer. Let us see the question. Let us see the answer. Thinking process. Water is needed to the every living beings, whether it is a unicellular or multicellular. Every organism require water, and without water we cannot sustain our life. Water is called elixir of life. Elixir of life means that is one of the most important thing. Without it we cannot live. So can you imagine that, can we live without water? Not because, not about the plant, about the human, we cannot live without water, right? Now think about the solution. Let us go to the solution part. Plant use water for the metabolic processes as the photosynthesis, transpiration and respiration. Every mechanism require water. Photosynthesis, water is a raw material. For transpiration also, and for respiration also you require water, and that is why some water comes out as a waste in the form of respiration as well. So plant die when water in excess, because excess water removes the air trapped between the soil particle. The plant require air also. Yes, the plant require air also. So air, you say that air is present in the outer atmosphere, so the shoot part can take. But what about the root part? Root also require air, and and that air which is trapped inside the soil particles, soil, between the soil particles the air is present, so that air is taken by the root surface. So that air is removed because you have given so much water to it between the soil particles. And you must be knowing this, that the water which is available to the plant is called as capillary water. The capillary water that is present between the soil particle. But if the water is present, so much water that soil particle is present in the water, then that is not going to help to water. There is no air to breathe the roots. So plant root do not get oxygen for the respiration process. And once root cell die, water and mineral absorption is stopped, leading to the gradual death of the plant. So this lead to the death of the plant as you water so much, right? Next question is here. What is the difference between the lenticels and stomata? Very, very important question. lenticels and stomata. Difference. So this question is again coming from the NCERT section 6.1. Let us see the answer of this question. Difference between the lenticels and stomata is as follows. So here the column wise difference we can see here, and the figure also. Lenticels form the loose epidermal tissue. It is a loose arrangement of the epidermal tissue. But stomata is a modification of epidermal tissue. The main difference. Here, there is a loosely arrangement of the epidermal tissue forms the lenticels. There is no modification, but there is just loose arrangement of cells. But see, in the case of stomata, epidermis modified to take a structure like a guard cells, bean shaped, and that forms a pore like structure that helps into the gaseous exchange. Okay, this is regarding the structure. Now lenticels, where they are formed, location found on stems, no guard cells. They are not having any guard cells in the case of lenticels, and they are found on the stem. Even the peel of the vegetables, the peel of fruits, they are having these lenticels. Okay. Stomata found on leaves has guard cells. Now, if you compare it with the stomata, now they are having the guard cell, but the lenticels are not having any guard cells. Openings not regulated. Here, the opening is not regulated. That they want, whenever they want to close the lenticels, they can close. Whenever they want to open, they can open. No, such thing never happens in the case of lenticel. So, it is not regulated. Now, coming to the stomata, regulated opening for the gas exchange. And why it is regulated? Because this guard cells do contain chloroplast and they contain chlorophyll inside it, right? So they can do the process of photosynthesis. They can synthesize glucose material in the guard cells. So that helps to regulate, because if the more glucose will be there, synthesize more glucose, then it can take water from the surroundings and they swells up, turgid condition. So they get open, right? And when the stomata are not having much glucose, so it means the photosynthesis is affected. So at that time they closes. So in that way, it is regulating the gaseous exchange, stomata. Next question is here. Write the precise function of a sieve tube. Yes, you can say that lenticels are open permanently. They are not regulated. So if it is formed, it will be open, right? Okay. Coming to the question, write the precise function of A, sieve tubes, B, interfascicular cambium, C, collenchyma, and D, arenchyma. So you have to write the function of these. Let us see the answer. First one is the sieve tube. So it is present in the phloem tissue. Yes, it is a part of the phloem tissue, a type of complex one. So its function is to transportation of synthesized foods throughout the plant. Yes, it is helping in the transportation of the synthesized food. And what is the food? Glucose. But in which form it is transported? Sucrose. So sucrose is transported through the sieve tube. Okay, through the sieve tubes. So here you can see the sieve tube element. This is the sieve part and this is the sieve tube structure. So from these, the sucrose is transported from one cell to the another, right? Next is interfascicular cambium, B1. Let us see the function of it. Its function is to bring about secondary growth in the dicot stem and the root. Yes, we have seen just now that the two types of cambium are there, interfascicular and intrafascicular cambium. So they are related to the secondary growth in the dicot stem and root, because the monocot is not having the any cambium, so no secondary growth we see in the case of monocot plant, right? It is secondary meristematic tissue present in between the two vascular bundle. Between two vascular bundle, I'm not saying that in vascular bundle. In vascular bundle means between the xylem and phloem, that is the cambium, and that cambium is called as intrafascicular cambium. And interfascicular cambium means between two vascular bundle. So this type of cambium originate at the time of division season, at the time of the season that you see that there is a continuous growth in the wood. So there the parenchymator cells transformed into the intrafascicular cambium and form a continuous ring like structure. Okay. So here you can see these, the bundles are arranged circularly. So between the two vascular bundle you can see there is a line, and that is the cambium. So the cambium in between the vascular bundle, in the vascular bundle, so that is called as intrafascicular, and between the two vascular bundle the cambium ring, that is interfascicular cambium. Next is the collenchyma, where we see this collenchyma. Its function is to provide the mechanical support to the young growing herbaceous system. Yes, they provide the mechanical support to the plant, and its cells have the angular thickening at the corners. Yes. So here you can see this is the collenchyma, and you can see the thickening at the angles. These are the angles of the cells, and there you can see there is a lot of thickening, right? So these are the thickening corners. Next is the D1, arenchyma. Arenchyma. So it provides buoyancy to the hydrophytic plant. Hydrophytic plant means the plant that we see in the case of aquatic environment. Aquatic environment. So those plants are called as hydrophytic plants. So it is a specialized parenchyma having large air spaces. They are having large air spaces, right? And it is a modification of parenchyma. It is these type of parenchyma is a modification of the parenchyma only. So when the parenchyma is having a lot of air spaces, it is called as parenchyma, right? So it provides buoyancy to the aquatic plant, and it can float on the surface of water because these parenchyma get filled with the air and makes the plant light so that they can float on the surface of water, right? So it is a specialized parenchyma having large air spaces, and the figure also you can see. These are the air spaces. Lacunae, it is called as lacunae, or the air spaces. Next question, and this is again short answer type of question. The stomatal pore is guarded by the two kidney shaped guard cells. Name the epidermal cells surrounding the guard cell, and how does a guard cell differ from an epidermal cells? Use a diagram to illustrate your answer. Very important question, and this question is coming from the NCERT section 6.1. Let us see the answer. The epidermal cells surrounding the guard cells of the stomata are called as subsidiary cells. Yes, we have guard cells, bean shaped guard cells, and there are some cells that surround these guard cells, and those cells are called as subsidiary cells. Differences between the guard cell and epidermal cells. So guard cell and epidermal cell, what are the difference between these two? So guard cells, they are the bean or kidney shape. If you see the shape, regarding the shape, then the guard cells are bean shape or a kidney shape. But epidermal cells, they are barrel shape. You know barrel, wooden structure in which the wine is made, or it is a block or a container, right? So that is a barrel. Shape wise, they are barrel in shape. And regarding the guard cells, if they possesses chloroplast, so yes, they possesses the chloroplast. But what about the epidermal cells? The epidermal cells are not having the chloroplast. They are not having any chloroplast. So they cannot do the process of photosynthesis. But yes, the guard cells can do the process of photosynthesis, right? And if you see the guard cells size, then they are smaller in size, but the epidermal cells are quite larger. Cell wall of the guard cells are not uniform and thicker. They are not uniform. The cell wall, they are the outer wall of the guard cell. It is thin, whereas the inner side it is thicker. This we have discussed. Epidermal cells, they are bigger in size and they are uniformly thin. They are not very thick, they are uniformly thin. So uniformity regarding the cell wall we see in the case of epidermis cells, whereas in the guard cells, the outer cell wall, outer side of the guard cell wall is thinner, whereas the inner side it is thicker. So the structure you can see here, right? Epidermal cells, see the epidermal cells, they are larger in size, and see the guard cells, they are very small, right? And they are having the uniform epidermal, this cell wall, whereas the guard cell, you can see the outer side it is thin, whereas the inner side it is thick, right? They are having the chloroplast, they can do the process of photosynthesis, but the epidermal cells, they do not have any chloroplast. Barrel shape and they are bean shape. So these were the differences. Next question. The point out the differences in the anatomy of leaf of people. People leaf, that is called as ficus religiosa, and maize, that is called as zea maize. Draw the diagram and label the differences. Let us see the answer. So people is a dicot plant. So we show the characteristic feature of the dorsiventral leaf, whereas the maize is a monocotyledonous. So leaf will be isobilateral. Discuss the anatomical features both leaves by comparing the difference like epidermis, stomata and vascular bundles. So this is the thinking process. And now we will see the solution part. So here is the differences. Character of the ficus leaf, dicot leaf, they are position wise, they are dorsiventral. Dorsiventral, they are having the dorsal surface, they are having the ventral surface. But the maize, it is called isobilateral. Isobilateral symmetry they are showing, right? Stomata wise, more on the lower epidermis. In the case of dicot, the stomata are confined on the more on lower surface of the epidermis, whereas in the case of monocot, the stomata are present equally on both the surfaces, equal on both the epidermis. So this is a very important difference. Mesophyll wise, palisade and the spongy parenchyma. Mesophyll, they are having the two types of mesophyll cells, column wise and very compact cells, they are having the palisade type, and the spongy, you can see the circular or oval type of shape cells, right? Those are called as spongy parenchyma, and they are having a lot of air spaces in between or the air cavity, right? So they are mesophyll. Now if you see the maize leaf, Only spongy parenchyma, no palisade type of parenchyma is found in the maize leaf. Major differences regarding the mesophyll. And you can see the air cavities. More air cavity is present in the dicot, whereas less air cavity is present in the monocot. Bundle sheath. Regarding the bundle sheath, parenchyma cells with some collenchyma, that forms the bundle sheath. And here the parenchyma cell with sclerenchyma, that the sclerenchyma forms the bundle sheath here, right? Bulliform cells. Bulliform cells are totally absent in the case of ficus, that is the dicot one, right? But in the case of maize, maize is a monocot, so they do possesses some of the bulliform cells. Next question. Palm is a monocotyledonous plant, yet it increases in girth. Why and how? Let us see the answer of this question. It is a monocot, and generally we see that the monocot do not show any secondary growth. So palm show the secondary growth despite being monocot. Growth occur by the division of parenchyma cells in the ground tissue. So you can see here, parenchyma cells are dividing in the ground tissue. Increase in girth through the repeated cell division is called diffused secondary growth. Diffuse type of secondary growth. Here, the parenchyma cells are there. Lateral meristem. Lateral meristem is not the correct answer here, because lateral meristem is the cambium, and cambium is not found in the case of monocot. But here, in the case of palm, they show the increase in the girth. So this increase in the girth is due to the division of the parenchyma cells in the ground tissue. So that increases the girth. And this type of increase in girth is called diffused secondary growth. Okay, so last question. Distinguish between the following: exarch and endarch condition of the protoxylem, stele and vascular bundle, protoxylem and metaxylem, intrafascicular cambium and interfascicular cambium, open and closed vascular bundles, stem hair and root hair. Let us see the answer. This is a long question. Exarch versus endarch. Protoxylem toward the outside and metaxylem toward the inside. That is common in the case of roots. And when you see the endarch, here the protoxylem toward inside and metaxylem outside, just opposite. And that condition you see in the case of stems, right? Stele versus vascular bundle. Where you see the stele? What is a stele? Entire central cylinder, including all vascular tissue, right? That is a stele. And vascular bundle, individual strand of the xylem and phloem, that is called as the vascular bundle. Protoxylem, metaxylem. Protoxylem first forms xylem. This we have already discussed. Smaller cells and thin walls. Whereas metaxylem later forms xylem, larger cells and thicker walls. Interfasciculum, intrafasciculum. This we have already discussed. Interfascicular cambium between the vascular bundles, and intrafascicular cambium within the vascular bundle. Open versus closed vascular bundle. Open, that contains the cambium and allows the secondary growth. Whereas closed one, no cambium, secondary growth is not possible. Stem hairs and root hairs. Stem hairs, modified epidermal cells, protective in function, and root hairs, single cells that helps in absorption of water. So this is all about today's session. Thank you very much for attending today's session. We will meet again. Till then, take care and bye.